Hello, my S2S students. Today we are going to explain the last activity in chapter 4, the genotype. This activity is divided into 3 parts. This video explains the first part. Let's start with some definitions. When you see the exclamation mark, this means that you have to memorize the information represented in the slide. The first definition to be studied is the gene. Do you know what is a gene? A gene, if you remember, is a DNA fragment that occupies a well-determined locus or place on the chromosome and that codes for a given polypeptide or a given protein. As you can see in this picture, you have 2 homologous chromosomes, one from the mother and one from the father. And on the 2 chromosomes, you have different genes, gene A, gene B, gene C, D, E, and F. Each gene occupies a given place called locus on the chromosome. For the gene A here, you have black color and light pink color. These colors mean that for each gene we have 2 copies or 2 alleles. So the second definition, allele is a version or copy of a gene. We take one allele from the mother and one allele from the father. So one gene is expressed in 2 copies within an individual. However, it is not necessarily that the gene has the same alleles on both homologous chromosomes. For gene D, we have same alleles since we have identical colors. Same as for gene B. While for gene A and gene C, gene E, we have different colors on the homologous chromosomes. The colors mean that we have different alleles for the same gene. So the alleles can be identically inherited from the mother and the father, or they can be different. Polymorphic gene. Poly means many, morphic, morphs, or form. We have seen this definition or this term in the previous activity when we talk about polyallelism and polymorphism, and we said that polyallelism, the presence of many alleles for the same gene, induces the polymorphism, which is the occurrence of 2 or more phenotypes in a population. A polymorphic gene is a gene that exists in the form of multiple alleles in the population. For example, the gene for hair color, the gene for eye color, the gene for hairstyle, and the gene for the ABO system, the blood group system. You have allele A, allele B, allele O, and we have different phenotypes, group A, group B, group O, and group AB. So the polyallelism or the presence of polymorphic genes induces polymorphism, the presence of many phenotypes, the diversity in a population. What is genome? Genome is the whole set of genes found in the nucleus of a cell. So it's the whole set of genes carried by the 46 chromosomes in the nucleus of a human cell, for example. Phenotype, as you know, is the all visible or hidden characters expressed by an individual. Like I have said before, example, the blood group A, blood group B, concerning the skin color, the hair color, the eye color, etc. So you have to study these definitions. We continue in the definitions. We arrive to the genotype. What is genotype? Do not confuse between genome and genotype. Genome is the set of genes carried by our chromosomes, while the genotype is the combination of alleles carried by an individual. So, if you can observe in this document or picture, you have two homologous chromosomes, one from mom and one from dad. And we have three genes, gene A, gene B, and gene G, but we have six alleles. Allele small a, capital A, allele capital B, allele small b, and allele G. So here we have two identical alleles. Here we have different alleles carried by homologous chromosomes. So this combination of alleles constitutes the genotype of an individual. The combination of alleles for all the genes carried by all the chromosomes. The genotype, for example, for the blood group A, an individual may have as genotype either AA or AO. This means that the individual may inherit one capital allele A from the mother and another allele A from the father, or he or she can inherit an allele A from one parent and an allele O from the other parent. But in both cases, for the both genotypes, we have the same phenotype, which is the blood group A. We will see later uh, why these two genotypes, these two different genotypes, give the same phenotype. We will discuss this information later. So for the genotype is the combination of the all alleles for all genes in the nucleus. Now, what is homozygote? Homozygote is an individual having two identical alleles for the same gene. For example, here, the individual in this document is homozygote for the gene G, since he or she has two identical alleles, capital G, carried on the homologous chromosomes. Another example, for example, the individual having as genotype AA for blood group A, so the individual will be homozygote. Homozygote means having two identical alleles on both homologous chromosomes for the same gene. Now, what is heterozygote? Heterozygote is the individual having two different alleles for the same gene. Here, this individual is heterozygote for gene B and for gene A, since he or she has different alleles, capital B, small b for gene B, and small a, capital A for gene A. Another example, for example, blood group A, an individual may have as genotype AO, so he or she has two different alleles of the same gene, which is the gene for blood group. So in this case, when the two alleles are different, we said that the individual is heterozygote. We say that the individual is heterozygote. Now, what is recessive allele? A recessive allele is an allele that is only expressed in the homozygous state. For example, allele O in the blood group is a recessive allele. Once found with allele A or allele B, it could not be expressed. We only have the expression of allele A and we obtain group A. For the allele O to be expressed, it must be present in two identical copies, OO, in order to obtain the blood group O. We will explain this later in the following slides. So the recessive allele, since it is not expressed in the heterozygote state, it is symbolized by a small letter. So here we can say that the small b here is recessive with respect to the capital B here. Same as for the small a here, this allele is recessive with respect to A. That's why it is represented by a small or symbolized by a small letter. And in this case, only allele capital A is expressed in the heterozygous state. Small a cannot be expressed here. Same as small b, which is recessive, cannot be expressed. It is recessive with respect to B. So B and capital A here are dominant alleles. They are expressed in both homozygous and heterozygous states, and they are symbolized by a capital letter. So big B or capital B is dominant over small b, so it is expressed in the heterozygous state, and it is also expressed in homozygous state. Now, one phenotype may correspond to several genotypes, as we have seen previously. Blood group A, which is the phenotype, corresponds to several genotypes. For example, here the genotype AA, which is different from AO, these two different genotypes give the same phenotype, which is group A. So how can we explain this fact? The ABO blood group system is defined by the presence of markers called antigens. Markers means proteins expressed on the surface of the cell and constitute its biological identity. So the red blood cells of our blood group system, the red blood cells express on their surface proteins or markers called antigens or agglutinogens. We have antigen A, we have antigen B. The expression of these antigens is at the origin of the following four blood groups. So, according to the combination of alleles and the expression of these antigens, we have four different groups: group A, group B, group AB, and group O. These four groups are determined, these phenotypes are determined by the presence of three alleles of the same gene, which are the allele A, dominant allele A, the dominant allele B, and the recessive allele O. Now, according to the combination of these three alleles, we have one of these phenotypes. So how can we explain that the blood group AB exists, but the blood group AO does not exist? When alleles A and allele B, allele A and allele B, when they exist together, they give blood group AB. While when allele A is found with allele O, we only have the blood group A and not the blood group AO. So how can we explain this? Allele A is dominant with respect to O. O is symbolized here by a capital letter, but it can be also symbolized by a small letter since it is a recessive allele. So some references symbolize allele O with a capital letter, but it is recessive. So allele A is dominant over allele O. That's why in the presence of allele A and allele O, the latter is not expressed. Only allele A will be expressed at the surface of the red blood cells, and we obtain antigen A, which is a protein, and this protein, its expression will give the phenotype, which is the blood group A. You know that one gene or allele codes for a protein by translation, and the expression of the protein gives the phenotype. So when allele A is present with allele O will not be expressed. Only allele A is expressed. It gives the antigen A, which is a protein found on the surface of red blood cells. Thus, we have blood group A. Same as for allele B, when it is present with allele B is dominant over O. That's why allele B will be only expressed at the surface of red blood cells. We have antigen B and blood group B. Now, since both alleles A and B are dominant, so when they are found together, they are co-dominant, which means they are both expressed at the surface of red blood cells. So A is expressed and B is also expressed. We have two antigens, A and B, on the surface of red blood cells. Thus, we have the blood group AB. So that's why we don't have the blood group AO. We have blood group AB. Since A and B are both dominant, they will give two antigens, A and B, that are both expressed on the surface of red blood cells. Now, when allele A or allele B are found with allele O, A or B alleles are only expressed, O is recessive, so we obtain either antigen A and blood group A, either antigen B and blood group B. So as you can see here, when a person is of blood group A, the red blood cells of this individual present on their surface the antigen A here in pink color and having a circular shape. So this is the antigen A. For blood group B, we have another type of antigen called antigen B, having a triangular or rhombus shape. So this is the antigen B. If we are of group O, there is no antigens, neither antigen A nor antigen B. So no antigens are found on the surface of red blood cells of group O. However, for group AB, since A and B are co-dominant, they are both expressed. That's why on the surface of this red blood cell, you see both antigens, antigen B in green and antigen A in pink. So in this case, we have a blood group AB. Now, to explain the presence of antigens on the surface of red blood cells, we must study the genotype of the individual. If we have a homozygous individual AA having same alleles, which are the alleles A here and alleles A here, so we have surely the expression of antigen A and we are of group A. And if allele A is found with allele O, since allele O is recessive, so only allele A will be expressed. So we also have antigen A and we are in group A. Now, if A and B are found together, they are co-dominant. They will be translated into antigen A and antigen B that will be expressed on the surface of the red blood cell, and we obtain the group AB. Now, when allele B is found with allele O, or when we have two identical B alleles, we obtain group B, since the red blood cell will present on its surface the antigen B in both cases. Now, when we have allele O, which must be expressed in the homozygous state, we must have two alleles O in order to obtain the blood group O. Since allele O is present, we don't have allele B, neither allele A, so we don't have antigens A and B on the surface of the red blood cell. So the red blood cell of group O doesn't present an antigen on its surface. Now, give the possible genotypes that exist for a person of blood group A and for a person of blood group B, as we have seen in this document. What could be the different genotypes? For blood group A, the individual may be AA or AO, same as for blood group B. So the answer is a person of group A may be homozygous AA, you can say homozygous, or heterozygous AO. Same for the person of group B. Now we are still in the blood groups. We are going to talk about the Rhesus system, Rhesus positive and Rhesus negative. So like the ABO system, the Rhesus system is based on agglutinogens or antigens. Agglutinogens is the same as antigens present on the surface of red blood cells. So we have antigen A or antigen B on the surface of red blood cells, but also we have other type of antigen called the Rhesus antigen or the Rhesus factor, here a triangular shape in green. So the individual with the red blood cells that have the Rhesus agglutinogen or antigen on its surface, they belong to the Rhesus positive group. For example, an individual may be of group A positive, B positive, AB positive, or O positive. This means that, for example, for A positive, the individual is of group A, he or she has antigen A on its surface, but since she is positive or he is positive, this means that near the antigen A, we find the antigen Rhesus. Now, for the people who don't have the Rhesus agglutinogen, so they are called Rhesus negative. Rhesus negative means that there is absence of the Rhesus factor. So the individual may be group A negative, B negative, AB negative, and O negative. For example, if the individual is of group A negative, she or he has the antigen A, but he or she doesn't have the antigen Rhesus. That's why she or he belongs to the group Rhesus negative. So this is what was the part 1 in this activity. In part 2, we will be discussing another polymorphic gene called the MHC gene. And in the third part, we are going to explain the biological identity and the techniques that allow to study the genotype of an individual. Thanks for listening.